All right, so welcome to week 4, online introduction to psychology. So today we're going to spend some time talking about sensation and perception. In the last lecture we talked about the basic neurobiology, and today we're going to expand that a little bit and talk about how our body processes sensory information and how we take that sensory information and we turn it into representations of the external world. So, generally speaking, the distinction between sensation and perception is sensation is just the process of receiving environmental input, and perception is organizing and interpreting these information, and creating meaning, creating a representation of the outside world. One important theme for this week is this idea that sensation is not the same as perception. One thing we will find as we go through this week's information is that two people can see the same exact event and perceive it in a completely different way. And this is in part because reality is inherently subjective. When you observe individuals behaving, your observation, your perception of that event is a combination of the environmental input, which you're actually seeing, but also your interpretation of that event. So in general, our sensory systems have specialized cells hello there, thank you, Java. So in general, our sensory systems have specialized cells whose function is to take external stimuli and convert that information into neural impulses, right? So last week we learned about this idea that the brain is this electrical chemical organ, and the basic structure in the brain is a neuron, and a neuron fires these neural impulses. Patterns of neurons combine together to form neural networks that produce all of the complex thoughts and behaviors that we have. And we talked a little bit about this when we listened to the podcast about You Are Here, where they talked about how the different neurons in the brain activate to give us a sense of where we are in space. One of the things that's sort of interesting about our sensory system is that, if you think about any given moment, your sensory systems have just a ridiculous amount of information that they're processing, right? There's the light, there's the sound in your environment, there's, taste, temperature, pressure, the feel of your socks on your feet, the way that the inside of your mouth tastes, how sort of dry or moist your nose is, right? All of those things that I just mentioned, the socks on your feet, the taste inside of your mouth, the level of dryness in your nose, all of these things you were not consciously aware of until I mentioned it, right? When I said, what does the inside of your mouth taste like? You can think, like, I'm chewing gum right now, so my mouth tastes minty. But unless you are actively thinking about that, you are not really consciously aware of all of that environmental input. And one of the other themes for this week is that sensory systems, they serve this sort of data reduction function. Our sensory systems take environmental inputs and organize it and reduce it down and condense it so that it's only serving up the information to our conscious awareness that is important and relevant in that moment, right? It may not be relevant to you right now what the inside of your mouth tastes like. So your brain filters that information out of conscious awareness. So our sensory systems serve this sort of data reduction function. Data reduction, this can happen in both directions. So this can happen both bottom-up. Bottom-up processing is processing that our sensory systems do, that our brain does, that's based on environmental input, that's based on our sensory receptors. And then there is top-down processing. And top-down processing is based on experience and expectations. Top-down processing is your stored representations working on the sensory information that's coming in. So the example that I like to give is when I was younger, I was working at a Pizza Hut, and I was a manager at this Pizza Hut, and I'm standing behind the counter, and all of a sudden this man runs into our restaurant and says something to me that at the time sounded like I didn't understand what he said. He ran behind the counter and immediately picked up the phone and got on the phone. Right? Now, it's generally unusual for someone to run behind the counter. Right? And as I sort of stood there in a brief daze, all of a sudden I realized what he had said was, there's been an accident, I need to use your phone. Right? So this is an example that illustrates how both bottom-up processing and top-down processing converge to create this sensory experience, to create this representation. My brain heard enough of what he said that my top-down processing could fill in the gaps so that I could then understand what he had said. Okay. Here's another example. So just take a minute. If you got a notepad in front of you, take a look at these little dots and just write down what you see. So you just take a minute, look at the dots, just write down in your notes, what do you think this is? Right? What do you think this is? Okay, once you've had a couple things, things do you think this could be? The next thing I want you to do is with the PowerPoint open, let's see if this works. Oh, thank you, QuickTime, my computer. Next thing I want you to do is you can click on this link. This should take me directly to this website. Excellent. Now what do you see? All right. Most of you, if not all of you, all of a sudden see that this is a person, that these dots represent a person walking. Okay. Now, if you want to mess around with this, you can make this more of a female walk versus more of a male walk. You can make the person sort of overweight, or you can make the person lighter. I guess this person's running. I don't know. So the important point of this demonstration is this idea that when you just look at these dots, right, the sensory information is not enough for you to understand what's going on. Once you see the motion of the dots, your brain recognizes that motion. It says, I've seen that pattern of motion before. That looks like someone walking. And then all of a sudden you see someone walking, right? So the bottom-up processing is the sensory input. That's just the dots, or the dots moving. The top-down processing is your brain taking all this sensory information and comparing it to your stored representations and coming to the solution, oh, this is a person walking. So we have these 5 sensory systems, and I'm going to go through and I'll talk about each of these in turn. So we're going to start with vision. Vision, as with all sensory information, we need to take external stimuli and transform it into neural impulses. And this process is called transduction. When we're talking about the visual system, we're talking about the process of transforming light energy into neural impulses that can be then read and interpreted by the brain. So in your environment, there is light bouncing off of everything. And that light is bouncing around the room, bouncing around wherever you are, and much of it is going into your eye, right? And the function of your eye is to focus and transform that light into neural impulses. So we'll talk about focusing. The structures of the eye, moving from the outside in. The cornea is just sort of this transparent layer over the eye. On the inside of this is the pupil. This is just the hole, the hole through which light enters. The iris is a muscle that controls the size of the pupil. If you know anything about photography, this is kind of like the aperture of a lens. Essentially, the iris will adjust the size of the pupil. They'll adjust the opening to either allow in more light or to restrict light. Right? So if you've ever, been out it's late at night or you've been in a really dark room for a while, your pupil will open up, and your iris will physically make the size of that opening bigger to allow more light into your eye. Okay, but the cornea, the pupil, the iris, and the lens are all structures that are designed to get light from the outside world into your eyeball. The lens is a flexible structure behind the pupil that actually focuses light onto the retina, which we'll talk about here in a minute. The lens itself actually changes shape, right? This is how you can focus on things that are far away or things that are close to you. The muscles actually control the shape of the lens, and they focus whatever you're focusing on onto your retina. Specifically, we'll talk about the fovea of the retina. Now, many vision problems that people experience are typically problems of focusing, right? So if the idea is to focus the light on a specific area in the eye, if the eye is not shaped properly, if your eye is too short, then you will be farsighted, right? So what happens is your lens focuses the light on a point that is actually behind your retina. And so this makes things look as if they are out of focus. The opposite side of this is if your eye is too long, you're nearsighted, right? So if the distance between the cornea and the retina, the back of your eyeball, is too long, when the lens focuses the light, it cannot focus it to the back. It focuses slightly in front of the retina. And again, this is nearsightedness. Okay, this is one of the reasons why not all people's vision problems can be corrected by laser surgery. Astigmatism is a problem with the shape of the lens. So the lens is misshapen, and that's why it's having difficulty focusing. With laser eye surgery, what laser eye surgery literally does is it, for lack of a better term, it burns the lens of your eye. It uses a laser to reshape the lens in your eye so that it focuses properly. But if the problem with your vision is the size of your eyeball, it's too short or too long, right? Obviously, this laser surgery doesn't work. Interestingly, as you get older, the lens gets less flexible, so you have difficulty focusing. Also, interestingly, as you get older, the lens starts to yellow a little bit, and this actually can affect your color perception. So someone who's very young and someone who's very old could look at the same color and see slightly different hues. Okay. So this is all about getting light into the eye. Once the light is into the eye, the next phase in this process of transduction is for the light to activate neurons on the retina. Now the retina, the way to imagine the retina, imagine that the retina is kind of like a carpet. Think about a carpet. It's got lots of little individual fibers, and this carpet sort of blankets the inside of the back of your eye. And in this carpet, there are little cells that respond to light. We'll talk about those cells in a minute. Okay, so embedded within the retina are a whole bunch of cells that respond to light. Now, the fovea is the very center of the retina. So the fovea is a special spot on the retina, and it's the very center. So whatever you're staring directly at, if you can imagine sort of the very middle of your focus. So if you put your finger out in front of your face and you stare at it, I mean, not like right in front of your face, but out sort of, arm's length out in front of your face, and you stare at it. The light that is bouncing off the tip of your finger as you stare at it is landing on your fovea. Okay. It's sort of the main focal point of the eye. Now, interestingly, your eye also has a blind spot. And this is a place in your eye where the optic nerve leaves the eye. So essentially, all of these specialized neurons that respond to light, which we'll talk about on the next slide, they send their axons through the retina, and all of those axons get bundled together to form the optic nerve, and the optic nerve carries these neural impulses to the brain, right? And in the spot in your eye where your optic nerve actually leaves your eye, you have a blind spot, right? There are no light-sensitive cells on that spot because it's all just axons leaving your eyes. So you may be asking yourself, if I have a blind spot where my optic nerve leaves my eye, why don't I see that blind spot? And the answer is top-down processing. Your brain takes information from surrounding cells in your retina, and it fills in the blind spot in your eye. Okay. Now, this isn't to say that you can't mess around with your system. So if you take your finger and you put it sort of in the corner of your eye, kind of like right where you can sort of just the edge of your, as I'm saying this, I'm doing it, right where the edge of your eye socket is, the bone, and you just push in on your eye, just push in towards your nose, what you will see is a little black spot. So if you take your finger and you put it in the corner of your eye and you push your eye in just a little bit, and again, you want to push your eye towards your nose, you will see a little black spot. And that little black spot that you see is your blind spot. That is the area of your retina where the optic nerve leaves your eye. Okay. Now, in the retina, embedded within the retina, so again, if you imagine the retina as a carpet, each fiber in the carpet is a photoreceptor. It is a specialized cell that responds to visual information, that responds to light, that is activated in the presence of light. There are two kinds of neurons, or these two kinds of neurons, photoreceptors, rods. These are sensitive to light and dark. They're primarily active. They're primarily active in low light situations. So if you've ever, sort of woken up in the middle of the night and gone to walk somewhere, you may or may not have noticed that your vision is primarily black and white. It's hard to see color in very, very dark, low light. And part of this reason is because when you are in very low light situations, your rods are the thing that are responding to that low level of light. Okay. About 120 million of these disperse throughout the retina. So again, we're talking about a lot of cells here. The cones, on the other hand, so again, two kinds of photoreceptors. Cones are sensitive to bright light, colors, and detail. So cones are most active in bright light. Now, what happens actually is that when you are in a very bright area, so it's sunny out, you don't have sunglasses on, the cones, which are sensitive to low light, get essentially bleached out or washed out. There's too much light, they become overstimulated, and they just stop responding. Okay? And in that scenario, in those situations, your cones are doing all of the transduction for your visual system. The light is going in through your eye, it's being focused on your retina, and these cones are taking that light energy and turning it into neural impulses that can be interpreted by your brain. Okay. Now, if you walk into a pitch black room from outside, where it's super bright, you walk into a pitch black room, you will see nothing, right? Because the cones are active and your rods are all washed out, right? But over time, your rods will start to come back online and your ability to see in that dark environment will actually improve, right? So this is why it takes some time for that to happen. We have a lot less cones, and they tend to be clustered near the fovea, which makes sense, right? They're sensitive to bright light, colors, and detail, right? They're going to be near the main focal point of the eye. Okay. One interesting hypothesis about eye patches in pirates is that pirates used eye patches to keep one eye sensitive to night vision. So one hypothesis is that, pirates back in the day, when they were sailing on ships before the advent of electricity, if they had to go below deck, many of these ships had multiple decks, there was no light down there. And so one hypothesis is that pirates kept an eye patch on one eye to keep one eye sensitive to light and dark, to essentially keep the rods going in that one eye. And then if they had to go below deck, they could just flip the eye patch to the daylight eye, and then their night vision eye would be working well. Okay. Kind of interesting. Okay. So once your rods and cones have converted that information into neural impulses, those neural impulses travel out of the back of the eye through the optic nerves, as you can see this here, through the optic nerves, and then it crisscrosses right here. And this is called the optic chiasm. This is just the spot in your brain where the optic nerves crisscrosses. So the nerve from your right eye crisscrosses and goes to the left side of your brain. The nerve from your left eye crisscrosses and goes to the right side of your brain. It goes through the thalamus, and the thalamus, which we'll talk about, is just essentially the main hub of sensory information. This is where all sensory, it's like a relay station of sorts, where all sensory information coming in and motor information goes out, all runs through the thalamus. At the end of this pathway is the occipital lobe. This is the back of the brain. This is where all of these neurons from your optic nerve terminate. And then they start working their way forward, well, in increasing complexity as you start to develop representations of, what's happening in your environment. So this visual cortex, it starts with these very basic rudimentary features of the environment. And it takes this very rudimentary information and combines it together to present a representation of the world to our conscious awareness. So for example, there are some neurons in the visual cortex, neurons and clusters of neurons, that respond only to specific features in the environment. So perhaps a cluster responds to just straight lines, horizontal straight lines. So one thing that we can do is we can surgically implant an electrode into a cluster, a very specific cluster of neurons. Typically we do this with animals because most people will not allow us to surgically implant anything into their brains. And then what we can do is we can present the animal with images, lines, corners, patterns, these kinds of things. And we can determine that certain cells respond to certain elementary features in our environment, but only those features. Right? So here we've got the cell's responses. This group of neurons goes crazy when it sees a rectangle in this orientation. When the orientation changes, it still responds, but not as vigorously. And then when it changes again, it's not responding at all. A brief note about animal models. We in research often have to rely on animal models to do this kind of basic research about biological function. There are review boards that are actually more strict for dealing with the care and research on animals than they are with doing research on people, significantly more strict. And so, although there are always instances where people are not following the rules, that there are always instances where animals are mistreated, for the large proportion of research facilities, the animals are treated with a great amount of care and in as humane a way as possible. So even though it's kind of freaky to see a monkey with an electrode sticking out of his head, you have to imagine that the monkey was probably anesthetized. The monkey probably isn't really aware that it's got an electrode sticking out of its head, and the like. Also, the monkey was probably bred for use with research. I like to tell students there are catalogs of lab rats that you can buy with whatever feature you want. You want a lab rat that's predisposed to get breast cancer, so you can do some breast cancer research, you can buy that rat. You want a lab rat that has a completely sanitary gastrointestinal system, so you can study probiotics, you can get that rat. So these animals are often bred specifically for research purposes. Okay. Now, another interesting thing about the visual cortex is that the visual cortex engages in parallel processing, right? The visual cortex is processing all of the sensory information that it's getting from your eyes at the same time, right? So this is movement, color, depth. It processes your body's position relative to your head, right? So when you turn your head to one side, right, that information is being processed at the same time as the light entering your eyes, at the same time as the sound entering your ears, all of these things. Now, there's a couple of theories about visual processing. So one thing that we know about color processing, one thing we know about cones in the retina, is that there are specific cones in the retina that respond to either red, green, or blue. And if you remember your elementary school art, you would know that red, green, and blue are the three primary colors. And with those three primary colors, you can make any other blend of color. People who are colorblind tend to lack one or two types of cones. So up here is an example. If you are colorblind, depending on what kind of color blindness you have, you may not be able to see the number in this field of dots. So if you're looking at this and you can't see the number 8, you may be colorblind. So trichromatic theory says that we have these cones that measure color, that respond specifically to red, green, or blue, and this is where we get color processing. Opponent processing theory suggests that neurons process opposing colors in pairs. Okay. So the idea here is that when you see, for example, some red in your environment, the green neurons that would respond to green, are also slightly active. The same thing is true with yellow and blue and black and white. Right? So you perceive red, but the green neurons are still kind of active. So they're processed together. So here's an example of this. Let me see. Negative after image. Oh, this is a great one. So I would click on this. It may not come through on the video. So what you need to do, oops, here, let me put this in the middle. So what you need to do is stare directly at the dot in the middle of the screen. And what you'll see happening is at first, when you look, you'll just see the pink dots, right? If you stare at the cross in the middle of the screen, eventually you'll see a green dot moving around. Now the green dot evidences opponent processing theory, right? Because there is actually not a green dot, right? This is just the after image of the opponent process. Essentially, the pink dots are activating, or the pink is activating the red cells. Those red cells are also activating the green cells, and then that's why you see the green dot. Now, what's crazy about this is that if you don't move your head, if you keep your head perfectly still and stare at directly at the little cross in the middle of the screen, especially if you don't move your eyes as well, eventually then the red dots will disappear. And essentially what's happening here is that the neurons that are processing that color in that field of gray are getting overstimulated, right? They can only fire their action potential so many times. They get overstimulated, and then you cease to perceive that pink. Okay. So these are the two theories about how we see color. Two main theories. Oh, wait. Oh, wow. Look at that. All right. So we talked about the visual system. We'll talk about auditory, and then we'll talk about touch, smell, and taste. So sound. Sound is kind of an interesting sense. In order to understand how we hear sound, you have to kind of know a little bit about how sound works. So sound itself, the sounds that we hear are actually waves of pressure that are moving through a solid, liquid, or gas. Okay. So if you've ever heard that in space no one can hear you scream, this is actually true. In space, there is a vacuum. There is no solid, there is no liquid, there is no gas. There is no substrate for oscillating waves of pressure to travel through. Therefore, there is no sound. So sound is created by some vibrating source. If you've ever seen like a giant subwoofer, you know, if one of your friends has a really cool sound system in their car, and they've got a giant subwoofer in the back, and you watch it, you'll see that the speaker itself is vibrating, right? And these vibrations of the speaker create that sound. It's that vibration that creates the sound. Now, once you start varying the properties of these sound waves, this is what determines things like loudness and pitch and timbre and all these kinds of features of sound. So if you can imagine a sound wave, it's just kind of like a sine wave you have here on this diagram. The intensity of the sound is determined by the amplitude. And the amplitude is the distance from the top of the wave to the midpoint, right? The higher the amplitude, the louder the sound, right? Frequency is the number of oscillations that occur in a fixed period of time. So an oscillation is going from the top of the wave to the bottom of the wave, back to the top of the wave, right? The fewer oscillations that there are in a fixed period of time, so you can talk about maybe oscillations per second, the fewer that there are, the lower the sound, the lower the frequency. The more that there are, the higher the frequency, right? And this determines pitch, right? So the super subwoofery, I don't know if subwoofery is a word, but right, the really sort of deep bass tones have very long frequencies, right? There are these huge waves and then maybe 3 or 4 cycles per second. Whereas really high pitch noises have many, many, many cycles in that same period of time, right? So amplitude determines loudness, and then frequency determines pitch. So the ear then is the sensory system that is designed to capture these sound waves and convert them into neural impulses. The ear has many different parts. We'll go through each of them. So there's the outer ear, which essentially functions to funnel and concentrate sound. So one thing that you can do, your sort of ear flap that is on the side of your head, it functions to funnel sound from your environment into your ear canal. One thing that you can do is you can change your perception of sound by just moving your ears. So if you grab the back of your ears and you pull them both forward a little bit and kind of pinch them together and pull them forward, you'll notice that your perception of sound actually changes. Right? The middle ear is the portion of the ear that actually physically transmits these sound waves from the outside world to the inside world. All right. So here's a close-up of the inner ear. The inner ear contains the eardrum and the bones of the inner ear. The eardrum literally is like the head of a drum. If you've ever seen a drum before, it's got sort of a skin wrapped over the top of it, and when you hit it, the vibrations that they create the sound of the drum. And the eardrum is much the same way. It's a thin membrane, and when sound waves come into your ear, they vibrate that membrane, and that vibration gets transmitted from the outer ear to the inner ear via the bones of the middle ear. Right, so the hammer, anvil, and stirrup, they have different, more technical names. They function to actually transmit the vibration, mechanically transmit this vibration to the inner ear. Okay. Now, this gets transferred to the oval window, which is actually just another membrane that takes these vibrations that are transmitted from the eardrum, from the eardrum, and transmits them into what we call the basilar membrane, the cochlea. Right? The inner ear is made up of this cochlea, right? And the cochlea is sort of a fluid-filled, snail-shaped structure. And the way to kind of imagine the cochlea is that within the cochlea, there's what's called the basilar membrane. Now, the way to imagine the basilar membrane is imagine sort of a field of wheat. And the basilar membrane is kind of like a field of wheat, with each sort of wheat stalk as a separate auditory nerve fiber, right? And what happens is that as those sound waves travel through the cochlea, they actually sort of sweep across these hair cells, these auditory hair cells. And it's this process of sweeping across these auditory hair cells that activates those hair cells and transforms that physical energy of waves traveling through space, sound waves, into neural impulses, right? So imagine when you're sort of standing and looking at the field of wheat and the wind blows, you can kind of see that blowing wind ripple through the field. This is kind of what's happening on the inside of your ear, literally right now while you're listening to me talk. Okay. So these hair cells actually generate action potentials. Now, there's a couple different theories about how our brain recognizes sound. So if you take a look at this image, we've got this cochlea right here. And the cochlea in this image, you can imagine it's a snail-shaped structure. The basilar membrane runs up the middle of it. But you can imagine if we could unroll this and straighten out the cochlea, we would see that, we could have sort of the basilar membrane represented as this long stretch of tape or field of wheat, if you will. And what place theory suggests is that different sound waves activate different areas of the cochlea, right? So high amplitude sound waves, they have lots of cycles per second. Those high frequency sounds are activating hair cells towards the front of the cochlea, and those low frequency sounds, right, with long frequencies, few cycles per second, those are activating the hair cells at the back of the basilar membrane. Okay. So this is place theory. Another theory is frequency theory. And frequency theory states that it's not about necessarily where on the basilar membrane the nerves are being activated, but that the brain actually interprets the rate of firing for the neurons in terms of the frequency of the sound, right? So the idea is that if you have a sound, let's say it's coming in at 100 hertz. Hertz is just a way that we measure sound. The auditory nerve, the basilar membrane, is sending out action potentials at that same frequency, right? At that same rate. If we increase the frequency of sound, then we're going to increase the rate of firing of these action potentials, right? So when we increase it to 200 hertz, it's firing faster. Okay. Generally speaking, frequency theory and place theory are both correct. One thing that is sort of a problem for frequency theory is that neurons can only fire so fast, and then at some point they top out. And so frequency theory does a really good job of explaining how we hear high frequency sounds. Frequency theory does a really good job of explaining how we hear low frequency sounds. Place theory does a better job of explaining how we hear high frequency sounds. And in reality, it's probably a combination of both of these things. Okay. If you are interested, if you're interested, you can click on, back here. If you're interested, you can click on this link right here, and this will take you to a website where you can determine the highest level of, the highest frequency that you yourself can hear. One thing that we learned is that as we get older, our hearing gets worse and worse. As we get exposed to loud or high-pitched sounds, our hearing can also get worse and worse. So hearing loss can be caused by damage to the mechanical system, right? So this is why you don't jam Q-tips into your ears, right? If you had a Q-tip in your ear and you accidentally fell into the wall and it got jammed into your ear, it would physically break and damage your eardrum and the bones of your inner ear. And this would prevent your middle ear from transmitting that information from the inner ear, from the outer ear to the inner ear. The other kind of hearing loss is sensorineural hearing loss. And this is damage to the receptor cells of the auditory nerves themselves, right? So this is when you've been, you're at the rock and roll concert, or you're standing behind, a jet airplane as it's about to take off, and you have this sort of prolonged exposure to high intensity sound that just physically damages the neurons, okay, the hair cells in the basilar membrane. Okay. Generally speaking, it's hearing loss, sensorineural hearing loss is a combination of exposure and frequency, or exposure and intensity. So the longer the exposure, the higher the intensity, the more the damage. If you listen to a super loud song on the L for 10 minutes, that's not as bad for you as listening to the same super loud song for an hour on the L. Okay. So I have a link here in case you're interested that'll take you to some interesting information about hearing loss.